Hello again. We have already talked about the basics of immunity in an earlier video. So this video is going to go over the two immune responses that your immune system provides you. They are humoral immunity and cellular immunity. Cellular immunity, sometimes called cell-mediated immunity. Humoral immunity is sometimes called antibody-mediated immunity. Now, before we get into the two immune responses, I want to review the importance of receptors on these cells and how the T and B cells become stimulated. Notice the dendritic cell, an APC, in purple. In both pictures, in the small picture, that cell has come in contact with a pathogen. And notice that the pathogen has a little structure on it, which is called a PAMP, or pathogen-associated molecular pattern. It's the little red ball that looks like a nose on its face. And on the APC, there will be a structure called a PRR, which stands for Pathogen Recognition Receptor. It's a special receptor. It identifies the PAMP, and as a result, we have activation of the APC, the dendritic cell in this case. It phagocytoses, takes it in, takes a piece of that antigen, a little antigenic determinant on it, that little red ball that you see, sticks it on the outside of its cell membrane with an MHC class II receptor. Remember, MHC class II are found on dendritic cells, macrophages, and B cells. That's the extent of the cells in your body that have that receptor. Now look at the big picture. You see that same APC, it's taken in a virus in this case, presents to a T helper cell. Notice the receptor attachments between the two cells. So let me go back and remind you about something. Here are two pictures of receptor attachments between the T helper and an APC. Remember that it is a dual activation system. The main receptor binding is between a TCR, T cell receptor, and the MHC. I've already mentioned usually a class II. That's where we're going to start. Not always though. Then you see also another receptor on the T helper called a CD4, or if this were a cytotoxic T cell rather than a helper, it would be a CD8. It co-attaches to the MHC receptor. Now, you also see another couple of receptors paired together, a CD28 and a B7. This is called a co-stimulatory receptor binding. It is the second signal. Remember that the purpose of this dual recognition system is to control the activation of your immune system and your T cells. In other words, there has to be a really good reason for the activation of your T cells. This is not something to be taken lightly, because remember, your immune system can be a big problem for you. It's not always good. So to take things seriously, we have to have a second signal that is involved. That then will cause the T helper cell, in this case, to divide into two possible subsets, a T helper 1 or T helper 2. T helper 1 is going to be most involved in cell-mediated immunity and activating the cytotoxic T cells. This is most important, again, for controlling cancer cells, killing them, as well as destroying viral infected cells. In other words, antigens, pathogens inside of your host cells. If we get a T helper 2 cell, we are going to activate humoral immunity. What will start this off would be an antigen like a bacterium, a protozoan, a worm, a fungus, something that arises outside of a cell, not a virus. This will be involved with activating T cells. So this is a busy picture, but to point out here that T helper cell up there that is talking to the APC can be involved with turning on cellular immunity, which is the box I've outlined in red on the right, or it can be involved with activating humoral immunity, which is the box I have outlined in red on the left. So the helper cells are pivotal immune cells that are involved in both types of immune responses. You will see this in more detail in a second. So let's start with cell-mediated immunity or cellular immunity. We start out with, again, either a cancer cell or a cell infected by a virus. There can be other conditions that start this, but these would be the two usual. Or what may start it would be an APC that has picked up an antigen and expressed it on an MHC class 1. Now remember, MHC class 1 proteins are on all of your cells, liver cells, kidney cells, any cells except red blood cells. So if a liver cell is infected by hepatitis B virus, and the hepatitis B viruses are being produced inside of that infected cell, that infected host cell in your liver will express viral proteins on the outside of its cell membrane to alert your immune system. And those viral proteins will likely be attached to MHC class 1. So, however this starts, let's go over to step 2. The T cells become activated, and they now can either physically attach to and activate other helper cells or cytotoxic T cells, or maybe even B cells. This can be done by physical attachment and recognition. It can also be done by chemicals. Easier to do it by chemicals. Remember cytokines? A subset of them is called interleukin. There are many of them, over a dozen at least. Some inhibitory, some stimulatory. And it's far easier for these chemicals to have an effect on cells when they are not in the close vicinity to come in contact with the T cells. Now, from here we go to routes possible. These turned on T cells, in the case of the antigen being extracellular, like bacteria and protozoa and others, will be presented on MHC class 2, and those T cells, when activated, will talk to B cells to stimulate them, turning on humoral immunity. We'll get to that in a bit. So again, step 4, not really part of cell-mediated immunity. I just wanted to point out that this can happen, but it's part of the other immune response. So step 3, the APC has presented to a T cell. The T cells now become activated, the T helper. They then can activate other cells, They can also become memory cells. You can have helper memory cells. You can have cytotoxic memory cells. They're to help with future immunity. They become activated much faster when they come in contact with the same antigen another time, second, third, fourth, and so on. So let me show you this on a picture. You've got a T cytotoxic cell in green at the top. Look at the frames on the right hand side. Well, actually, look at the left. You've got a cytotoxic T cell that has now been programmed and activated to recognize a viral infected cell, which has a certain viral protein that has been expressed on the outside of it in green. I wanted you to notice the receptor attachments between these two cells. Those receptors are all important in this system. Now go to the right. At the top, you see a very common system that cytotoxic T cells use to destroy the viral infected cells. They produce a protein called perforin, small purple cylinder structures. They embed in the cell membrane of the viral infected cell, creating a pore. At the same time, the cytotoxic T cell is making a protein called granzyme. Granzyme now moves through the pores into the viral infected cell. The viral infected cell now is activated to produce chemicals that cause it to commit suicide, to be destroyed. That is called apoptosis. There is a second mechanism by which cytotoxic cells can destroy viral infected cells. You can see that at the bottom. It basically works the same way, but some different structures that are involved with the recognition of the viral infected cell. And here's another picture on the right hand side that's showing this. Again, there is not one exact mechanism. The antigen may be presented to a T helper cell. The T helper cell may then talk to a cytotoxic cell. The antigen may be presented to a cytotoxic cell directly. One point I wanted to make here is that if the same antigen gets into the body again, the same virus, months, years later, there is a little bit of a change that occurs, and that is only one signal is needed. The binding of the TCR receptor and the MHC class II with the viral protein, that's the only one needed. There is no dual recognition because that's already occurred earlier on the first recognition. So this means that the process of cellular immunity can kick in much faster. Now let's turn over to the B cells. On the picture, APC is in purple. Let me jump down to the yellow cells, which are the B cells. Now in this case, on the picture, the APC is presenting to a T cell helper, and the T helper, which is about in the middle of the frame, is the one that is talking directly to a B cell, presenting its own receptors, different receptors by the way, for recognition of the B cells, and you see the antigen there in orange. The T helper does not have to be involved. In fact, there are two important things here. The APC may directly present to a B cell without having to go through a T cell. Second, there may not even be an APC involved. Remember that B cells can be phagocytic. They are not your usual phagocytic cells like dendritic or macrophages, but they can phagocytose, and so they can actually self-activate after phagocytosing. They do not need the APC. They don't need the T helper. That's usually the usual way that it happens with those cells, but not always. When the B cell becomes activated, it produces daughter cells. Some of the daughter cells become memory cells, memory B cells. And again, those memory B cells may become activated at a later time, months, years in the future, with the same antigen, and they will become activated much faster and more daughter B cells produced upon second and third and fourth exposure. Now, the plasma cells. Some of the daughter cells of the activated B cell will enlarge, mature, and become plasma cells. The big cell, in fact, the biggest cell on that page, is the plasma cell. It has a lot of endoplasmic reticulum, lots of ribosomes attached to the ER. They are manufacturing units for the production of antibody, and you see those antibody molecules being excreted. They make one type of antibody. They're not going to make a variation of IgA and IgG and so on. Only one type, and it's going to be very specific for that antigen. And remember that the antibody then goes out into the blood and will attach on to the antigen when it sees it again, making for a much better destruction of the antigen by phagocytic cells when they come in contact with them. So here is a look step by step of, I think, what I've just taken you through. In addition, as with the previous mechanism, cytokines like interleukins may be involved here in further activating other B cells to do the same thing. That transmission EM at the bottom right is showing you a plasma cell, and you can see the extensive network of rough ER with all of the ribosomes for the production of protein. Here's a nice comparison. You might want to stop here and look at it. I'm going to go on. And this is a nice graphic I found online. Maybe a bit busy, but does a nice job. Over on the left, you have the events occurring, starting with the dendritic cell, APC, coming in contact with the pathogen. But over on the right, the top part of that graphic is showing you cell-mediated immunity, and at the bottom on the right, you see humoral immunity. Each one of these immune responses has a particular thing that it is involved with. For humoral immunity, its big job is to fight against acute infections that are usually going to be bacterial, like streptococcus, which causes strep throat, staphylococcus that causes toxic shock syndrome, or maybe it causes an abscess or a boil. Gonorrhea, caused by Neisseria gonorrhea, that's an acute infection. Remember, those are extracellular antigens. They are outside of your host cells. Humoral immunity is also involved with a type of allergic reaction called an immediate type hypersensitivity. Will be covered on the next video. And some autoimmune diseases are related to it. For cell-mediated immunity, two important things: viral infected cells and chronic infections. For example, there are a few bacteria, like the one that causes tuberculosis, the one that causes syphilis. Those cause chronic infections rather than acute infections. They last a long time. They're rather insidious, and those are best controlled by CMI. Another type of allergic reaction called delayed type hypersensitivity, some autoimmune diseases, transplant rejection, all mediated by cellular immunity and the control of cancer cells. The picture up at the top right is showing you cytotoxic T cells in gray trying to kill a cancer cell, which is the big cell that is in tan. And the picture at the bottom is showing you a nice interaction between T helper cells and B cells, as well as APC, in trying to destroy a cancer cell. Here's the last slide I want to pull up. It does a nice job of giving you the big picture, I guess. You may want to stop here and look at it a bit. And that is all we are going to cover for this video.